Good day to all, I am Candace Franklin and welcome to my gel electrophoresis lab. In this lab, we will cover what is gel electrophoresis, what are the underlying principles of why it works, how does this experiment parallel to DNA electrophoresis, how and when is DNA electrophoresis used as a biotechnological tool in biology, and share how this application can be used to help scientists. Gel electrophoresis is the process in which different DNA molecules are separated based on their sizes. Based on the charge of the DNA molecule, which is negative, with the positive side of the tray, we can see which molecules are larger or smaller. We can also see how fast the molecules move towards the positive side. The experiment I am conducting is similar to DNA electrophoresis because they both entail methods of separating molecules according to their size and charge, and they both use the gel and wells that the molecules can travel through. DNA electrophoresis is used in laboratories to carry out DNA and RNA analysis. It can help scientists to figure out the relationship between species by looking at their DNA molecules. Electrophoresis is also used in forensic science. For this experiment, my hypothesis is that hopefully all the colors will move eventually, and I think that the lighter colors will move faster towards the positive side rather than the darker colors. Let's find out if I was right. The materials you'll need to conduct this lab are 5 9-volt batteries, 2 alligator clips, positive and negative, assorted food coloring, baking soda, small cups or containers for the food coloring, 2 stainless steel wires, corn syrup, agar-agar powder, 1 pipette, plastic container, 1 pair of gloves, water, gel comb, and a piece of cardboard, as well as a beaker. Now that you know the materials you'll need, now we can start the experiment.
Once your agar-agar is dissolved into the water, it should look clear and not grainy or cloudy. Now we will pour it in to our container along with our gel comb inside. Once your liquid solution is poured in, it should look like this with your gel comb halfway submerged in the agar-agar solution. It should not be all the way down to the bottom because this will cause leakage. Now we will wait until the agar gel is firm to the touch. Take 2. Take this pre-made agar-agar, put it into a pot of hot water so that it can melt, and don't forget to take off the cover when doing so. Now I have my agar-agar heating up on the stove at medium heat. In the meantime, as my agar-agar melts, I'm going to be making my buffer solution. So that is 1 cup of water and 0.2 grams of baking soda. Once your agar-agar has been cooling down for 10 to 20 minutes, you can now pour it into your container. Once you've put in your comb, your project should look like this. The gel comb should not be all the way down to the bottom, or else this will cause leakage.
While my agar solution is hardening, I will then work on my food coloring added with water and corn syrup. Now take 3 milliliters of water and put into each container. Then add 5 milliliters of corn syrup into each container. And then add in 2 drops of each color. And in the last 2, 1 drop of red, 1 drop of yellow, and 1 drop of blue, and 1 drop of red. And now we mix them. Now we have our 6 colors: blue, green, yellow, red, orange, and purple. You have mixed all your colors. Now you can check back on your agar-agar gel. And as you can see, it has set properly. Now you can take out your gel comb. And there are our wells. Now that your gel is all set, you'll take a sharp object or a knife to cut out the sides. Then you can take a spatula to scoop out the gel. And here is the result. Now you'll pour in your buffer solution that we made earlier and pour it into the container. Make sure that the gel and your wells are fully submerged into the water. Now you're gonna take your 2 stainless steel wires and put them into your container. Now with your 9-volt pyramid of batteries, you'll put the positive side farthest from the wells and the negative side closer to the wells. Do not connect the alligator clip to the next wire because then it will start the experiment. Now time to take your pipette and add in the coloring to the wells. First we'll start with blue. Put your pipette into the well. And then fill it. Next color, green. Next color, yellow. Next color, red. Next color, orange. Next color, purple. Now that you have filled your wells, you can now clip on your other alligator clip to the wire. It should start to bubble now. Whoa! Oh, it's bubbling. I made another diagram and I put which colors moved from the slowest to fastest, and the blue moved the slowest, then the purple was the second slowest, the green was third, the red was fourth, the orange was fifth, and the yellow was sixth, which means the yellow moves the fastest. That concludes my gel electrophoresis video. I hope you enjoyed.